Hello, everybody. Can you hear me today? Yay! Oh, my prayers have been answered. Oh, heavens. Okay, good. I'm glad you can hear me. Guys, hello, and welcome everyone to part 5, Understanding Gene Therapy and How It Pertains to SCN8A. My name is Casey Craig. You may know me as the SCN8A mom to Stella, but recently I've had the pleasure of volunteering for the Acute Syndrome Foundation by organizing the SCN8A Family Book Club and moderating all the clinical trial readiness educational series, except for the one that my audio didn't work. This 5-part series was aimed at helping all of us gain perspective and information as we prepare for the upcoming clinical trials. To sit down and have a casual conversation with industry professionals, clinician researchers, and advocates about important details regarding our children and their future. Wow, I don't know about you guys, but this has been an absolute delightful experience for me. Last week, we had the pleasure of chatting with 3 amazing panelists regarding the setbacks and failures in drug development. If you missed parts 1, 2, 3, or 4, no worries. The link is in our SCN8A Facebook group where you can go and watch those recorded videos. Now moving on to today's event, Understanding Gene Therapy and How It Pertains to SCN8A. Well, we have 2 wonderful panelists that are here to prepare to answer some of these prepared questions that we have for them. Some of the questions that they are going to address today were gathered this week from parents in the SCN8A Family Support Facebook group. But we also welcome you to, if you need any type of clarification or comments, to put those in the chat box, which I'm getting ready to tell you about now. All right, so a few housekeeping items. So group interaction through questions or comments or you need clarification are going to happen in the chat box. So if you'll notice to your right, there's a little area that you click on chat box and you can type in your questions that way. To your left, you'll also notice that there's a tab called Expo. In this area, you will find troubleshooting if you're experiencing technical issues, or you may also type a request for help in the chat box. Our fabulous technical guru, Megan Varner, will help you tackle any technical issues you may have today. Before we get started, I want to take a moment to give a very warm welcome to the Duke 15 community that are joining us as we partner together in this clinical readiness journey. I also want to extend a huge thank you to our sponsors for this event, Neurocrine, Praxis, and Zogenix. Now, I would like to introduce our very own Hilary Savoy. She is the director and founder of the Acute Syndrome Foundation and has poured her energy and focus into fighting for our SCN8 community for the past 7 years. Not for the title or the personal recognition, but because she's a parent just like us that wants nothing short for our children to be happy, to thrive, to have more good days than bad. On behalf of all our parents and our children, Hilary, thank you from the bottom of our hearts for your countless hours spent preparing and producing this educational series. You're a rock star mom, fellow parent, and leader. Without further ado, Ms. Hilary.
Hi, everyone. Can you hear me okay? Yeah. Thank you, Casey. You never failed to get me a little teary before I get to work here. So thank you, everyone. Welcome for joining us. It's just such a pleasure to be here with our SCN8A families. And as Casey said, I am the founder and director of the Acute Syndrome Foundation. And we're here today for our last session, which is going to focus on gene therapy. I'm going to take a little bit longer today to talk, just to sort of discuss some of the takeaway from this series before we get started. And also talk about what's coming up in lead up to our 6th annual clinician, researcher, and family gathering in December. So we may run a little long today in pure acute syndrome fashion. We put together this series in order to help provide insight into the drug development process, to share some thoughts about the roles communities like ours can and should play in that process, and to empower our families to make informed decisions as they consider the research and clinical trial landscape. I must say that I've been really pleased by the outcome of this series. The attendance, the conversations, and the engagement have been just fantastic around this topic. And it's been really a pleasure to have this informal space to engage really some of the most interesting and knowledgeable people I know working in drug development, advocates and clinicians with clinical trial experience, and researchers who are responsible for helping drive the future of cutting edge therapies. I've also been very happy to share my 5-week cold with all of you. So I'm sorry about that. As I've said, probably too many times, the people who we've had on this series are the same people who've taught me along the way. I've personally really never learned well in lectures. I learn in dialogue and human connection. And I know it's a little bit of a joke in our community that I've learned most of the science that I know over drinks. And it's a bit funny because it's true. Because the drinks after sessions were the places that I felt comfortable asking the kinds of questions that I worried were maybe a bit stupid, I guess. And even as I worried that they were a bit stupid, more often than not, they led to some really interesting and informative conversations because they got at the foundations of how all of this is done, how it works. And those were foundations I didn't have. So these are the conversations of the variety that I've wanted really to bring here for all of you who, like me, might learn best in this way when things just feel very human scale. My PhD is in communication and rhetoric, so I don't have a background in science. I do have training in trying to sort of sync up the needs and meanings of various groups. And this is why my passion with acute syndrome has always been in bringing parents and our children to the forefront of communicating with researchers, clinicians, and industry. There were obviously tons of research meetings that talked about SCN8A before we started our gathering, but what we've always done differently, right from Julianne and my first vision of our first gathering in Philadelphia in 2015, was that we wanted our families there, unmediated, speaking for themselves, interacting with the people who will change the course of our children's lives. We wanted our children there, making noise, playing on the floor, connecting by being themselves. I always thought that this was not an impediment to a scientific meeting, but rather that it could be the heart of it. And I think that's borne out. Last year at our gathering in Baltimore, a researcher who I don't know all that well came over to me at the gathering and said that this is exactly why she looks forward to our meetings so much. That it's that environment, the kids being themselves, the parents, the human scale. So as we gear up for our gathering this year in December, virtually, it of course makes me sad to not be able to share that same experience of us all being together in person. However, what I have loved about this year, and I was just talking to Miriam about, I haven't loved much about this year, but this bit is true, has been the new connections and the newfound forms of connections that we can build together as a community, like we did through our social and emotional support programs, funded with a grant from the Child Neurology Foundation and support from Neurocrin, this clinical trial readiness educational programming to really informally address clinical trial topics of interest to our community. Thanks again to support from Neurocrin, Praxis, and Zogenix. And then the upcoming reimagining of our annual gathering as a virtual event to bring more of us together. And this is funded by a grant support by NORD, sponsorship again from Neurocrin, Praxis, Zogenix, and Greenwich Biosciences. And I think also just the variety of support we've received for these programs is really telling. So I'm thrilled to announce that in the lead up to the gathering, we will be continuing these informal sessions, only now with some of the researchers and clinicians who will be sharing their work at the gathering. And these sessions will give you the chance to be up close and personal with the researchers as they tell us, the families, about their research ahead of the gathering in a space where you can ask your questions openly and easily and get prepared for the upcoming gathering in December. So starting in November 11th, we'll be joined by Drs. Ricky Moeller, Katrine Johansson, and Alina Gardella to discuss their 2 papers that they'll be presenting at the gathering, information on loss of function phenotype and information on sleep challenges in SCN8A, as well as other findings from their registry. And then on November 18th, we'll speak with Manoj Patel and his lab about their work. With that, to the topic at hand, gene therapy. We picked this as our final topic to end the current series on because there's really a ton of interest about gene therapy among our families and the general public. And there's so many different expectations about what gene therapy really is, and lots of mixed messages about how soon these therapies will be reasonably available. We feel as though it's kind of a basic primer on gene therapy in general, and gene therapy as it pertains potentially specifically to SCN8A would be incredibly helpful for us to manage our expectations, especially as we look toward at least one small molecule trial, while families may be wondering how soon after that gene therapy trials could be happening in our community. So, without further ado, to discuss this today, we have, and Miriam and Steve, if you can click on your join buttons, we have Miriam Meisler, PhD, who's a Myron Levine Distinguished University Professor, Professor of Human Genetics, and Professor of Neurology at University of Michigan. And Steve Petru, PhD, Director and Head of the Department of the Florey Institute of Neuroscience and Mental Health, Head of Ion Channels and Human Disease Laboratory, and Co-founder and Chief Scientific Officer of Praxis Precision Medicines. And here she is, Miriam.
Hi.
And here's Steve. Hi. So glad to see you guys here. Thank you so much for joining. As always, this is a panel discussion. We have a super casual conversation, although both Miriam and Steve are going to share some slides with us as well. So just a note to the families before we get rolling is, please understand as you ask questions, there may be details about current programs that our panelists can't comment on fully, and note that a lot about gene therapy is unknown. And so we're talking about looking into the future a bit here. So the questions we have may not all have answers, and the lack of answers can be important too. So with that, I'd like to ask each of you to introduce yourselves and give a little bit of background on your work and your relationship with SCN8A. And Miriam, if we could start with you, that would be great.
Sure. Hi. I'm delighted to be here, and thank Hillary for the invitation. I'm a researcher at the University of Michigan, and I've been working on SCN8A for 25 years, actually, mostly with mouse models, and then as the human mutations were discovered, with looking into exactly what the effects of these mutations are on the gene function. And we've also generated 2 lines of mice in which we've put patient mutations into the mice. And these mice, the seizure phenotype, and we've been able to ask a lot of questions about what exactly is going on using these mice. So that's my background.
Thank you. And Steve. Oh, I think you're muted.
Hi, my name's Steve Petro. I'm a professor of neuroscience at a research institute in Melbourne, Australia. And for the last 20 years of my career, I've been very much interested in genetic epilepsies, such as SCN8A epilepsy, Dravet syndrome, SCN2A epilepsy, and others. I'm very much interested in understanding how the genetic mutations that are either inherited or arise anew or de novo cause the disorders in the patients. Want to understand the disease mechanism. Then we also want to try and understand how we can do something about that. And that's why part of my passion has been also to try and work with companies, either trying to found them working with them to try and take that next step, if at all possible. Very much would love to end my career on a note of having had an impact on the patients in terms of a therapy and improving quality of life. And so that's been me, and I'll talk a little bit today around gene therapy.
Thank you so much, Steve. As I mentioned in my lengthy intro, I'm often a little afraid to ask silly scientific questions, but in my experience, those questions are some of the ones where you get at the bits that get glossed over. So, Steve, I'm going to start with asking you a really silly question. What is gene therapy?
Thanks, Hillary, for that. I have prepared a few slides, so I will looking for the little screen sharing thing.
Yeah, so right at the bottom there's the camera and then perfect.
Can you see that? Yep, I can see it, so presumably you can.
And people, you can double click on it and it'll make it a little bit bigger if you want to see up close.
Terrific. So in answering that question, it's important to understand how genes break in genetic epilepsy. And I've just shown a little scheme here in the human's journey from fertilization to development. And genetic epilepsy can arise either from an inherited change that's contained within the egg or the sperm, or it can, as I said, arise anew or de novo anywhere along this journey from the fertilized egg up in different points in development. And if it occurs early, the mutation will be carried in all the cells. If it occurs later in development, it'll only be carried in some cells. And you may have heard of that word called mosaicism. And that's because the mutation, when it does arise, and it can arise because a gamma ray from the sun happens to hit the DNA at that specific point in time, or some other event occurs, a chemical event that results in an error when that gene is copied, or a chemical event that causes a fusion of two nucleotides that then produces the mutation. And it's random, it's uncontrollable, and it happens to everybody all the time. And those small changes are what drive the process called natural selection in the human population and in all animal species, why we change and evolve. But it can also occur in the wrong part of the genome, and that's why we see these disorders arise. And the way, when you think about the sorts of genetic rearrangements that can occur, an event can cause a breaking of the DNA. Other events can occur where you duplicate, and I'll just mention a few of them here. A new single nucleotide mutation arises in the child at some point, and that can produce gain of function, which is very relevant to the SCN8A community. And that's where the gene works harder or does something new. Or there's loss of function, and that's also part of what happens in SCN8A patients. And that's where the gene either is completely missing, a copy of the gene, or it just doesn't work as hard or as well. And paradoxically, there are some cases where you can get a bit of both, where the gene might do something new. A sodium channel gene normally allows sodium ions, which is, sodium is part of table salt, to go in and out of the cell under controlled circumstances. But mutations can cause another ion to go in and out, such as a proton, which is the hydrogen ion that makes up acid, can go through. And even though the channel might also not work as well. So there's all combinations of functional change. And that's really important when we think about how do we treat the mutation. If it's gain of function, you might want to reduce the function. If it's loss of function, you might want to try and increase it. If it's loss of function that's also got some very strange new function, you might want to ablate that gene and potentially increase the amount of the working or healthy copy of a gene, bearing in mind that most of the de novo conditions that affect many of the patients in this community, whereas one copy is normal and one copy has the mutation, and that's important to understand. We can get changes in the number of copies of a normal gene, deletions and duplications of genes, whole stretches of our genome can be copied or deleted. We can inherit a copy of an otherwise not so harmful gene from each parent, and getting a not so harmful copy of a gene from mum, a not so harmful copy of the gene from dad, can cause what we call recessive disorders. And they're more common than we thought. Inherited mutations that affect the X chromosome. And this you don't see the disease appearing so much in the males, but you do see it in females. And there's a whole big part of the genome that's largely unexplored for its potential role in disease, and that's non-coding DNA. I mentioned genetic mosaicism, and epigenetics is where the environment impacts the gene chemically in a way that's inherited, but is not the traditional mechanism of point mutation. So there are lots of ways that you can break a gene to cause a disease. Predominantly for SCN8A, we're in category 1. And you'll hear a little bit from Miriam Miser, some very elegant work she's completed to try and therapeutically address that. And I'll talk a little bit about the whole concept of gene therapy. And as I said before, how it's broken determines how you think about going about fixing it. And then if it's a gain of function mutation, like you might see in many of the SCN8A patients, you might want to think, well, we might need to delete that copy of that gene or fix that copy of the gene. And that's where gene therapy comes in. And I'll give a broad definition. There are some narrow ones, but a broad definition is a treatment that directly operates at the genetic rather than the protein level. And here is a little cartoon of a gene, double helix, and at each location here where I'm showing, I don't know if you can see my cursor, at each location here is where a nucleotide sits that encodes the protein that this gene makes. Oops. And this is where mutations can arise. Now, I'll also include in gene therapy the idea that you can do this to DNA, but you can also directly try and treat the RNA. And that's the work that Miriam will show with ASOs. And I sometimes like to consider that holistically as gene therapy, RNA or DNA. Some people call that RNA therapy, but it's very closely related. That what they share in common is that they're a very precise way of trying to address the disorder. You're trying to directly target the disease mechanism at its most fundamental and early level. And we think that's important because, as we all know, when these mutations arise, we have multiple changes in the patients. It's not just seizures, it's other comorbidities that are as important or more important in some cases to quality of life than just the epileptic seizures. And these things arise because these genes participate in a whole host of physiological functions in the brain. And therefore, if we look at the symptoms and we devise therapies, which has been traditionally how pharmaceutical companies have addressed this by looking at, let's make a drug that stops the seizures, or let's make a drug that helps the movement disorder. When we look at it that way, we're only potentially addressing a part of the story. But when we think about gene therapy, and then even modern day therapy that's very targeted to the mechanism, we can start to think of fixing the gene and the dysfunction very early in the cascade. So all the emergent disorders that arise can be addressed at once. And that's really important to understand, because that is important to patients and families. Now, if within gene therapy, you can think of gene augmentation therapy. That's where you deliver a new gene to the body, or you turn a gene on. And there are ways that you can do this. You can package up a whole gene into a vector, and I'll give you an example of that in a minute. Or you can use specific mechanisms to turn a gene on and make it work harder and make it produce more copies of the protein that's necessary for function. You can do gene inhibition therapy. You can turn a gene off or turn it down, and we'll hear about that for Miriam in a second. Or you can do, and this is really what's new and on the horizon, is gene repair therapy, where you can actually go in and edit and genetically change and repair the mutation at the genetic level. And if you can do that, and the caveat is, if you can do it in every cell where that gene resides and is important for function, and not impact other areas of the genome and cause new problems, if you can do that specifically and accurately, you can then 100%, fix the genetic mutation at its most fundamental level. If you do it at the right time during the development of the child, you could almost consider that to be a cure. And you would have heard of that with respect to CRISPR, and I'll talk a little bit about that at the end. So there are all these ways, and each of these has got a different technology attached to them that lets you try and address these things. Turning genes on can use different technology to delivering a new gene. Editing genes, there are different approaches that people are considering. CRISPR is one of them. Earlier, there was another approach called zinc fingers that could do a similar thing. But CRISPR has opened up new ways of thinking about that, and you all would have heard about that. So, as I said, what's so good about it? Well, gene therapy targets the cause. If you have a gene that's broken, can you replace it? So in the case of loss of function mutations for SCN8A, and which presents clinically different to the gain of function mutations, you might think of, can we deliver a new copy of the SCN8A gene to the cells that need it? And that's the fundamental question you ask. And in the gain of function disorders, you might say, can we either ablate that gene or can we edit that gene to fix it? And if we ablate the gene, we then might be left with a loss of function phenotype. Do we then need to think of ways of turning on the remaining good copy or adding another good copy? So sometimes it might even be a two-step process. So I'll just give a really quick example from one of the examples that we've heard a lot about is spinal muscular atrophy. And it's caused, it's a movement disorder. We've all heard of Spinraza, which is an antisense oligo, and there's a new therapy on the horizon for that falls into that gene replacement category. I'll just compare and contrast those a little bit to try and explain the different modes of gene therapy. In this disorder, there is a gene called SMN1. That gene is either missing or not working in these patients. When that gene is not working, the protein it's meant to make, which is called the SMN protein, doesn't exist in sufficient levels, and those neurons will die. They don't function very well, and they die, and then they are not available to control the muscles, and that's why these children end up being wheelchair bound and not being able to move, because the neurons or the nerves that control the muscle are not healthy or dying and unable to work properly. Now, fortunately, so that this gene is missing, there's a related gene called SMN2. It's almost as good as SMN1, but there's a particular difference that makes it way less potent. When you make the SMN2 protein, it doesn't quite do what SMN1 does. Spinraza, which is the antisense oligo, converts the SMN2 into a form that looks more like SMN1, and it does that in a particular way that I won't go into the details of that. But what it does do, it increases the amount of SMN2 gene that's been modified, and that SMN2 gene can now take over the role of the SMN1. And we've heard of lots of wonderful stories about patients that have been treated Their movement has been improved. Some kids coming out of wheelchairs as a result of this. Now, there's a gene replacement therapy approach also being developed that's an alternative therapy for the Spinraza. And in that case, it was a simple idea, difficult to execute, but a simple idea that you can put a copy of a new version of the SMN gene into a thing called a viral vector. So a vector is what they do with viruses that are often the way that we get new genes into people is that we take a normal virus that can infect the body, can infect neurons in the body, and these normal viruses that like to get into neurons and make us sick. We can remove bits of those viruses that make us sick and let that normally are there for the virus to replicate so that it not only infects us, but it also makes more of itself so that we pass that virus on to other people. And we have no better example of that than the world we're living in now with COVID. That's what viruses do. They evolved to enter a host, replicate, and that host then makes more copies and then passes it on to somebody else. And the viruses that can do that better, those are the ones that survive more and are more prevalent in our population. But we can remove bits of those viruses, keep the good bits that we like, the bits we want. Well, we can use the viruses as a Trojan horse. We can attach a gene of interest to the virus, and then the virus, and give that virus, that modified virus to patients, and it'll carry a gene. Rather than carrying virus genes into the cell, it carries these new genes that we control into the cell. And there are limitations on the size of the gene you can put into a virus. Unfortunately, many of the sodium channels, such as SCN8A, are a little too big for some of the traditional methods of doing this. There are some workarounds around that try to overcome that limitation, and it gets very technical, but you can break the virus up into multiple virus copies, and then they can try and rejoin and make a bigger thing than they normally would. But the fundamental idea is you drop a new copy into this viral vector, this Trojan horse. You then the challenge, and I know this was in some of the questions that were asked, the challenge is, how do you make sure that you only get that into the cells that it's meant to go to? The body normally, we have in every single cell, we have the potential to express every single gene that's in every cell, but no cell ever does that. We have 20,000 or so genes that are available to be turned on, but in any given cell, you only turn on a fraction of those. But when you do this viral method, often you don't have any control of where it turns on. Every cell that receives the virus will express the gene. That, for some genes, that can cause a problem. And you don't want the expression of this gene to be everywhere. For SCN8A, for instance, if you made a version of the virus that could carry the SCN8A gene and you gave it and somehow delivered it to the brain, and then exactly how you deliver it to every cell, how you deliver it to the whole brain to get it broadly expressed in the areas that matter for this disease, are additional technical challenges. But if you can do that, how do you get it just to the cells that you need to get it to so that it fixes it? That's a challenge in the delivery. And there are methods that people are thinking about to try and address that challenge, but that specificity is very, very important. For SMN, it wasn't so important. And the idea is this is a new drug that's in very late stage development, and it's had clinical trials called Zolgensma. You may have heard about that. That drug, they did what I said. They packaged in the DNA. They removed DNA from the virus, and then they made the vector. Into that vector, they added in the SMN gene, and now it's available in a viral vector. They could infect patients with this. And the idea is the virus then gets in. This blue thing is the virus. It's in the neuron, and the blue little dots the new protein it's making, the SMN protein that was missing, that wasn't there in sufficient amounts. Whoops. And that new protein is what's giving life and making these neurons healthy so that they're available to control the muscles. And this will be a cure for these patients, because this virus will stay in the cell, and it'll stay and it'll continually produce protein for as long as that neuron is alive and the patient is alive. So this is a once-off treatment that could be considered a potential cure for these patients. Now, of course, once you've done this, there's no going back. So when you do gene therapy, and if in 6 years or 5 years or 2 years, there's a better approach for that, that option may and probably won't be available to those patients if they've already been treated with one round of gene therapy. And that's always something that's important to understand. And this is, I won't go into the details, but these are the stories of patients, and there are many of them with the Spinraza, about how beneficial it was to their lives and the new things that they could do. Not always 100% cure, but significant and major impacts on the lives of these children. That's been a really good story, brings a lot of hope to patients with genetic disorders around the world. And it's the sort of thing that drives me and Miriam to try and bring these sorts of concepts to patients with SCN8A, SCN2A, and SCN1A disorders. On the horizon, for the SCN1, for Dravet, they're thinking about, there's different companies that are looking at delivering SCN1A genes, which is missing in Dravet, for the common forms of Dravet. They use the specific, because SCN1A is a big gene like SCN8A, the sodium channels are all roughly the same size. They've got a various 2AAV approach to try and overcome that. We know that there are other antisense oligo approach methods that are trying to manipulate the amount of expression and turning on the existing gene more in order to overcome the lack of SCN1A protein in these patients. Again, these are ways of addressing the loss of function. For the gain of function, you can try and turn off the amount of gene that you express, and it's less clear. Classic gene therapy may be less relevant to a gain of function case, unless we use the gene therapy in the mode where it's trying to get in and cause repair. Well, I'll pass this data, but just talk about this idea. There's our copy. CRISPR is a gene editing technology, and this is a nice image from the web that I found that shows that CRISPR, in its perfect deployment, if you solve all the technical challenges around getting the vector that carries the CRISPR system to the right cells, making sure that you're removing the mutated part of the gene and you'll put in a blue copy right in this position and completely fix that. Once those technical challenges are overcome, we could then see, and that the nice thing about CRISPR is that it doesn't matter whether it's gain of function or loss of function, you can fix it. But it's not necessarily going to help those patients with really large duplications. And we know we have that family group with us today. And there are other approaches, because once you've got multiple copies of a gene in the system, CRISPR is not going to be able to repair that particular form of genetic rearrangement. Other approaches we talked about, and we'll hear in way more detail with Miriam, will be important to either replace a copy of a gene or to ablate the genes that are there in multiple copies that are causing the pathology. And you might need to think of ways of turning off multiple genes if you've got a large duplication in order to address the pathologies that emerge. And interestingly, and this is my last slide, that CRISPR system can actually be operated in a mode where you can even turn a gene on and you can turn a gene off with quite high precision. CRISPR is good because it can specifically target a gene. And once you've specifically targeted, you can carry activating or what we call repressing elements, along with that targeted interaction of the CRISPR. Here, we're not causing editing. Here, we're removing that editing aspect, but we are carrying along a new function, which is to turn a gene on or to tune a gene off. And these are other technological approaches that are on the horizon for gene therapy that could address not only classic point mutations, but also duplications. So I think there's, there are questions about maybe, when is this going to happen? Well, some technologies like antisense oligos are here and now. And I showed you that for SMN1 and Spinraza that hit the medical profession several years ago. We know Zolgensma, which is an AAV gene therapy carrying a fresh copy, has already been in trials, is on the verge to, it may have even launched, but is on the verge of launching. There were a few technical issues that hit that program very much in its late stage development. So these things are happening here and now for the very simple cases, and then there are many caveats as to whether a gene is a candidate for one approach or another, and what other. And I'll just end on a note to say one of the things that you always need to worry about with any sort of therapeutic intervention is what are the things that this therapy does that are good or efficacious, but then also very much keep an eye on what are the potential downsides of the therapy. What are the toxicological issues that can arise? And almost every therapy has that aspect to it. And it's the balance between efficacy and toxicity that there has to be struck just in the right amount so that we get all the benefits we need, and we can either live with the side effects or they're so small that we don't even notice them. And that's the challenge to all the companies that are trying to develop these therapies. They're trying to strike that right balance. Sure, we can use CRISPR to fix a mutation in the dish, but then moving that concept into a person has many new challenges, and that slows down and frustratingly for everybody, not just parents and patients and families and caregivers, but even for us as researchers. We'd of course like things to happen more rapidly, but we also need to be cautious and make sure we don't do more harm. And it's that balance of trying to be as fast as you can, deploy and understand the technologies, ensure that the very basic work that Miriam and I do, which is often way before the therapy arises, and that understanding keeps happening. We understand the disorder. We understand in 8A all the different ways that gene can be broken. We understand, and we can identify those patients. And so we can say, this subset of patients can have this type of therapy. This subset of patients can use this type of therapy. All those things are really, really important on the journey towards delivering a new therapy. I'll stop there, and if there are other questions, I guess we can try and handle those in question time. So thanks, Hillary. Long answer to a short question, I know.
No, that's perfect. Thank you so much, Steve. And this is just such a insightful presentation and so helpful, I think, for families to understand some of these distinctions. And so, Miriam, Steve talked about ASOs, antisense oligonucleotides, which I always stumble over. And he also mentioned your very elegant research on ASOs for 8A. Could you talk to us a little bit about ASOs and how, and your research how they pertain to 8A?
Right. Let's see.
You want to bring up your slides?
Trying, yeah.
Okay, so it's the third box over there at the bottom. There's the camera, the mute button, and then a monitor looking.
Yeah, I did click that, but it didn't bring my screen on. Let's see.
There's a little monitor with a diagonal line on it, Miriam.
Yes, I clicked that, and
did a
now
pop-up come up?
they're asking me
It then asks you which screen you'd like to share.
Choose what you'd like to your entire screen. I've clicked on that, but it hasn't shared apparently. So let me try that again, but it's not happening.
Is the line still through the monitor?
No, the line is off the monitor. Now I get this thing that says, what would you like to share? And I click on your entire screen, but nothing happens on this page. Do our...
Megan, do you want to pop on and give us a hand? We finally get to see Megan.
So once you selected the box, and then do you see share and chat?
No.
Let's see.
It opens a window.
Yeah, there
Okay.
should be a window that pops up that says share your screen. And then when you click your entire screen, right under that to the right says share and cancel.
Yes, that's right.
Okay. And then once you click share, it should pop it in there.
Share is not live, and it's not responding when I hit share.
There's a possibility if you have a pop-up blocker that might be preventing it from happening.
Do you know where I would find that?
Oh, thanks for answering that question, Steve.
Without looking at your screen, it's hard to tell exactly. If you try application window and select the exact application you're trying to share.
So
Try and
it's
see.
PowerPoint, right? Did anything happen just then? No, I'm afraid I'm.
Do you want to email them to me, and we can speak more casually about them, and then while they come through, I can pull them up.
Oh, that's a simple solution. Yes.
The age of figuring things out as well.
Sorry, everybody.
Oh, no worries.
Right away.
Steve is answering questions about loss of function and gain of function in the chat, so that works out perfectly.
Okay,
I'll keep doing that.
Desktop.
Yep, no, this is the age of workarounds.
Okay, so now I'm going to.
So Steve, if you want to, do you mind answering that question out loud, just so everyone, in case they're not checking, while we're pulling up those slides? So I think that's a really important piece. Yeah.
Sure. So one of the questions was, how do we determine gain of function from loss of function? And what that can be done in these experiments that are called electrophysiological analyses. There's a couple of different forms of that. But if we have knowledge of the mutation, so when your child is diagnosed, you will get information about the genetic change. And if we have knowledge of that, we can genetically engineer that same change into a copy of the gene that we've got in a cell that we can keep in a dish, and we can make that cell produce that gene and the protein, and then we can actually measure its function with a little electrode that we put onto that cell. And then by measuring that function, we can tell whether it's gain or loss. We can get a lot of information about how that gene normally works as a so-called ion channel in NAV 1.6, which is what SCN8A encodes. And if it's changed, we can tell whether that change will cause a loss or a gain. And so that's relatively easy to do nowadays. Takes about a month or so from when you know the mutation to when the result emerges, but that might be something that's going to be necessary when thinking about which type of therapy the patients would qualify for.
And how can a person get access to that kind of testing?
So that's generally not available as something that can be ordered. What often, as labs that do this, we tend to get overwhelmed by the physicians that are doing the diagnosis for their patients, and they say, well, we've got a patient with this mutation, could you please analyze it? And we try to accommodate as many of those as we can, and there's enough labs doing this around the world that someone generally can try and help out and get that done. So, and it's going to become more and more important as therapies become available. That might be something that needs to be formalized in the same way that genetic diagnosis is. There might be a need to formalize the functional testing so that we can connect the patient to the right drug.
Okay, well,
Did you receive that, Hillary?
I'm sorry?
Did you receive the PowerPoint?
I did, yep.
Oh.
I just pulling
Great.
it up now.
Oh, you're wonderful.
Can you see it?
Brilliant. Thank you so much.
Oh, no problem.
And Steve, that was great. I really enjoyed that. That's a great picture of the whole picture of where we are. Now, I thought I would concentrate on the ASO approach to SCN8A because that is the one that is most accessible right now, has the most potential. Will I be able to advance these? I guess you'll have to, Hillary.
Just let me know and I'll click.
Next slide, please. Okay. So I thought I would start with the basics that underlies why ASOs work. And that is the fact that DNA is double-stranded and it's held together by hydrogen bonds, those verticals that you saw in Steve's figure that we're all used to seeing in this double helix. Now, if you heat that double helix, you can separate those two strands. But the remarkable thing is that they will find each other if you cool it back down. There's a great gain in stability when these molecules become double-stranded. And that's what we take advantage of with these little ASOs. So the next slide shows you that even very short oligos, now we're talking about 20 base pairs, which is really very little, they can still form stable double-stranded hybrids with large molecules. And we do this all the time. This is the way PCR works. You've probably all heard of PCR. These little 20 base pair oligos have sequence specificity. And if you heat up that double-stranded DNA on the left and make it single-stranded and then cool it, these little 20-mers will find their exact match in the DNA, and they will bind there stably. And that's the basis for what we do with ASOs. So if you look on the next slide, you can see that ASO oligos are also these 20-mers. The remarkable thing is that they are taken up by cells, and in particular by neurons. If you can get these ASOs into the cerebrospinal fluid, they are taken up and they will hybridize with single-stranded RNA molecules, which may be in the nucleus or in the cytoplasm. So on the left, in the nucleus, the ASOs will bind these pre-mRNAs that are not yet spliced. And that's the approach for spinal motor atrophy. You change the splicing and you can change the product of the gene. But in our case, we're targeting the cytoplasm. On the right side of this cell, you see mRNA. That blue molecule is mRNA. And if we get the right ASO in there, it will bind to its matching mRNA. And this is the key. There is a enzyme in the cell called ribonuclease H. And when it sees this little RNA DNA hybrid, it will degrade it. That's not a normal molecule for the cell. So if we can target the RNA with a complementary DNA, which is the ASO, then the cell will get rid of it for us. And we can reduce the expression of that gene. Not at the gene level, but what I call genetic therapy. It's not gene therapy because we're not touching the gene, but it is touching the RNA that is synthesized by that gene. So the next slide is a technicality. So we can skip that and go to the next one. So, as I said, the first application of ASOs is to decrease gene expression. Now, if we focus on SCN8A in the developmental and epileptic encephalopathy class, which is what I think most parents in this group are probably dealing with, these are gain of function changes in the gene, as we were just hearing about. So SCN8A encodes this major sodium channel, and you're all familiar with the phenotypes of this disorder. Now, the predominant molecular mechanism, every case that I'm aware of, the mutations are de novo. They haven't been seen in the parents, only in the child. They're all missense mutations, which means it's a simple change of one amino acid for another, and they're all gain of function mutations. Gain of function meaning that the protein is present, the protein is synthesized, but its characteristics are altered. And I have an example on the next slide of 3 categories of gain of function mutations that we've seen. And as Steve said, the only way to detect this, the only way to know whether your child's mutation is gain of function or loss of function is to do this kind of experiment. But as I said, clinically, these early onset epileptic disorders are all gain of function. Exactly what the gain of function is, you can see some examples here. For example, on the left side of the slide, the wild type channel is shown with its activity versus the voltage across the neuron. And the black, you can see the black rising as the neuron is depolarized. But the red channel, which is the mutant in these children, is opening earlier than the wild type channel. And this makes the neurons hyperactive. And this is why you're having seizures. The one in the middle, the channel is opening normally, the mutant channel, but it's refusing to close when it should. And that makes, again, too much firing, too active neurons. And in the third case, the channel is staying open too long. So on the top, you see that the wild type channel should open very briefly and close. These are amazing machines that open and close within milliseconds. But some mutations, like the one on the right, keep the channel from closing completely. And so you have leakiness, activation of the neuron, and it's ready to fire too soon. So those are 3 of the kind of gain of functions that you can find out by doing this electrophysiological test. Now, there are some large centers now that are systematically going through mutations. I think there are probably several hundred mutations of 8A out there, and about less than 100 have been tested, maybe 20 or 30. But there are some high throughput methods coming, and there's a center at Northwestern in particular that is gearing up to do this at a high throughput, because in some cases it's relevant to the drug targeting. It's certainly relevant if you have a loss of mutation, but as I said, the loss of mutations of SCN8A do not cause seizures. So the next slide is the title of a paper that we published earlier this year, in which we were able to use those mouse models that I mentioned, where we have put the patient mutation in the mouse. And we were able to try out this idea about ASOs. And we did get very good protection in the mouse, SCN8A, and also in the, interestingly, in the Dravet mouse. So next slide. So here's our mouse. And I'm showing you the percent survival as a function of how old the mice get to be on the bottom. And we have in these mice early onset epilepsy and sudden death. And the mutation here is R 1872W. So you can see the black line at the top are the control mice, and they all survived this 12-week period. In red, you see the mutant mice. They have one copy of this mutation, and they have a second copy of the gene that's completely normal. And you see these mice only live for a few weeks. They have a very reproducible time course. They start having seizures, and they die mostly within a day. They die very rapidly. So the mice are much more susceptible to these mutations, and they do not survive. So the next slide shows you that we generated an ASO. The question was, can we treat SCN8A encephalopathy by reducing the expression of the NAV1.6 using an ASO to activate mRNA degradation? And here you can see where our ASO is. It's at the end of the messenger RNA called the 3' UTR. This is the 20 base pairs sequence of it, and it's been modified to make it stable in vivo. The next slide shows you what happened to the mRNA. So this ASO reduces the SCN8A transcript, which is another word for messenger RNA, in living animals in the brain of wild-type mice. So to answer another one of your questions, these ASOs do need to be administered into the brain, into the ventricles, by intracerebroventricular injection. And in these experiments, we injected all the animals when they were only 2 days old, because that's technically feasible. And it's before the onset of the disease. We're now doing experiments to confirm that this will work after the seizures begin. So here you can see 3 different doses of the ASO, and you can see with a 0, this is 100%, this is the level of messenger RNA in untreated animals, and with either 15 or 30 micrograms of DNA injected into the ventricles of the brain. As I said, the neurons took it up, it affected the mRNA level, and we got down to 50% of normal mRNA level. And this is turning off both that normal gene copy and the mutant gene copy. We're not specifically targeting the mutation because that's not feasible with ASOs. So if we look at the next slide, you can see how effective this treatment was in the mice. So on the left, we're again looking at the % survival as a function of how old the mice are. And you can see the mice that got this control ASO, the first line, look just like the untreated mice. They live for about 2 to 3 weeks. But moving to the right, the mice that got 15 micrograms lived for almost twice as long, and 30 micrograms even longer, and 45 micrograms even longer than that. So it was a very clear, unambiguous result. This really does work, and in vivo. And on the right, you just see that this is practically linear with the amount of RNA of ASO that you've injected. The more ASO, the longer they lived. But we couldn't inject any more than that. And so you see, this is about 9 weeks, which is 2.5 months in this case. But then we found what we thought, well, what if we give another injection, say right over here? We'll give the top dose, top tolerated dose, and then we'll give them another injection. And sure enough, that lengthened the effect. The next slide, Hillary, please. You can see here's our 30 micrograms. So we have the untreated, the 30 micrograms, and then when we gave another injection, we lengthened that even further. So we went from 2 weeks survival to more than 9 weeks survival. 2 times 4 is 8, 4 times lengthening. And this period further, they were seizure-free. There was absolutely nothing that we could detect in these animals until they began to have seizures. Their EEGs were normal, and their movement was normal. So let's look at the next slide. So then why do they then start to have seizures? They look fine for several weeks or months, and then seizures begin again. So we looked at the RNA again, and we asked, well, is that because the ASO is essentially no longer functioning and the RNA is accumulating again? And in fact, that is what happens. 3 weeks after the injection, you can see we're down to 0.5 times the wild type, or 50%. But by the time we get to 6 or 7 weeks, when they're starting to seize again, we're back to normal. So it's clear that we're going to need repeated treatments. And one of the parent questions was how often? Well, for that SMA example, the Spinraza example, it's roughly every 6 months that they have to again do a administration through the spinal cord injection. So this is temporary, but on the other hand, it also means that if you did run into trouble, you could stop it, and the messenger RNA would come back and you would go back to where you were. So the next slide. We were interested, since SCN8A is such a major driver of excitation in neurons, we thought that maybe this would be a general approach. And even if the mutation was not SCN8A, maybe this would be therapeutic for reducing excitation in other kinds of genetic epilepsies. So on the next slide, I'll just briefly show you that we tried it on the Dravet mice. And in this case, the lower curve shows you the normal course in Dravet mice. And we found, sure enough, that one dose of this oligo, this ASO, rescued these mice for up to 6 months. So we're now testing some other types of epilepsies as well to see if this might be a general approach. So then on the next slide, there's a brief summary. The 8A ASO directed to the 3' UTR of the message does reduce the mRNA in vivo, and the reduced expression rescues the seizures in the mouse model of SCN8A, DEE, and also in the mouse model of Dravet syndrome. So Hillary, if you could skip to slide 27, the rest of the slides I have are about treating Dravet by another approach, but I don't think it's really relevant here. So if we just look at this summary about the therapeutic potential of ASOs for genetic epilepsies, the positive feature is that they're tremendously specific. The sequence of that ASO will only bind to the RNA that it is complementary to, and you don't have to worry about cross effects on other channels the way you do with many of the drugs. The ASOs are actually very expensive to produce, and this may be this approach is broadly applicable to gain of function, and as I didn't show you in the Dravet case, to loss of function mutations. But on the other hand, the intrathecal administration is not the most desirable. We have not yet gotten the ASOs to cross the blood-brain barrier, although we're doing some trials with some methods that might solve that problem. Secondly, they have limited in vivo stability and need to be repeated every 3 to 6 months. And you definitely need to know that you have a gain of function mutation before you try to knock the messenger RNA down. So I'll stop right there, and we can move to questions.
Fantastic. Thank you so much. Let me just pull up my questions now. We got so many wonderful questions from the families. And I was able to share them in advance, everyone, with Miriam and Steve. And I know Steve's been answering some of the questions here, but we'll start with timelines. how soon could like the ASO treatment that you're talking about, Miriam, how soon could that be a reality for our kids?
I would say not within a year, not at least, for at least a year. There's work that has to be done with human targeting. This was a mouse. This is proof of principle, but we're not there yet. Unfortunately, I'm sorry to say.
No, I think I always find that our families are much more comfortable when they hear real answers, because I think it can be hard when hopeful answers and then those hopes don't come to fruition. Steve, what about some of the more one and done kinds of solutions that you were talking about? You know, how far out are things like that for 8A?
Yeah, I mean, I think there are programs that are looking at making small molecules that are specifically interacting with 8A, and you may have heard of those in some of your other talks. They're probably the ones that are closest on the horizon to treat the gain of function. Loss of function, 8A gene replacement, I don't know anyone who's working on that at a company at this time. There are other approaches that might also be trialed, again, with small molecule drugs that you can take orally as a pill that can turn these channels on. So the existing copy of the channel can be switched on a little bit more to try and overcome the loss of function phenotype. ASOs are obviously one of the hopeful approaches. And I know Ionis, who worked with Miriam and also worked with me on an SCN8A program, is interested in trying to deploy these ASOs clinically. And again, they have to achieve the right balance of safety and efficacy. It's something that's very much on their minds. So probably gene replacement therapy, could be the thing that will approach the loss of function patients. And CRISPR is the hope for all sorts of patients, not only SA and SCN8A, but a whole range. And there are companies that are still, really beavering away to try and get that done. We'll probably see CRISPR applied to cells that can be taken outside the body first, treated, and then checked to make sure that they're good, and then put back in. So that'll be important for blood disorders, where you want to take them out, do your genetic manipulation, and put them back in. I think that's going to happen. And I think once that starts happening and people understand, and then there are lots of efforts still on trying to make the CRISPRs more accurate. One of the big problems with CRISPR is that it doesn't only fix the mutation, but it's a little bit non-selective, and it'll cause random mutations elsewhere. And that might result in a tumor or something else from appearing. So people are very, very cautious there. So I think the gene therapy, in the way I explained it, is probably further away than it is for Dravet and for other disorders. But what is important is if companies, I think it's Novartis that's looking at the Dravet gene therapy, if they're successful, that success really spurs on people to think, well, can we apply this for loss of function SCN8A? Can we apply this for loss of function SCN2A as well? And I think so it's really important that we root for the success of these other programs, because that really breeds positivity in the drug discovery community, and then they can see a way forward.
If I could raise a question, Steve mentioned that for all of these approaches, you have to balance efficacy and side effects. And that is certainly relevant for an ASO to SCN8A, because if you go too low with your gain of function, you're going to have side effects. And this is something that I think is a real issue, and perhaps depends on the severity of the clinical syndromes that people are dealing with, whether one is willing to tolerate a specific side effect with the for the gain of stopping the seizures, basically, is something that will be an issue in the future, I think.
So those side effects would start to look like a loss of function phenotype?
Exactly. And two parents have mentioned that they seem to have LOF mutations with seizures. I think that's very rare, but I'd be interested in what those mutations are and what the evidence is that they're LOF.
Yeah, why don't we, the parents who have made those comments, connect up with me afterwards, and I will loop them in with you, Miriam, because I think that is the loss of function community is growing somewhat, and I think it would be very interesting to make sure that they are getting proper attention from a research perspective, and we will have the loss of function phenotype discussion coming up in November with the Danish epilepsy group. So that should be very interesting. And Miriam, actually, if you would like to join us, you would be more than welcome, of course. So I think that you read my mind on the risks question. I think that's really important. With the ASOs, I know we talk about with small molecule precision medicine that there's a hope, even though maybe the clinical trials would focus on seizure, that we would hope that perhaps we would also see improvement in some of the other comorbidities. Is that a hope as well with ASOs?
Oh, definitely. There was a question about the muscle effects. Definitely, these ASOs get taken up by the spinal motor neurons in the spinal cord and would definitely address the muscle problems and the ataxias. Yeah.
And I guess looking at some of the other parent questions from the group, would gene therapy be tailored to each child's specific mutation to block the gene with the mutation so that the other functional gene becomes dominant? Or could there be a single therapeutic that would work in many children?
So gene therapy would be, you wouldn't tailor it to a specific mutation, but you could tailor it to a particular class of mutations, such as loss of function, gain of function, as a group. So it's difficult to commercially develop for a single mutation. There are ways you can use ASOs in this, what they call allele specific mode. And there are ways that you can do that even though it can address multiple patients, even though their mutations are different, the ASO can still target that same copy. And that would be the next generation of the sort of molecule that Miriam talked about, that would only reduce the function of the mutated SCN8A and not also impact the wild type. And that's possible with an ASO, and you do this so-called linked SNPs. So with every mutation, there are other differences that make the mother's copy of the gene and father's copy of the gene different. And you can rely on those differences to target the ASO, and those differences are likely to be present in multiple people. So several ASOs, and I know Ionis is developing that technology for other disorders. We're looking at it for other applications in other genes as well. So that will be the sort of next generation of ASOs that will give some specificity, but it's probably going to be very unlikely that you'll develop a therapy for a single private mutation that might only be in one or several people.
Okay. I love that we are getting just so many questions here. Jinzia asks, preliminary data shows positive results for ASO in Angelman syndrome. Could this speed up ASO approaches also in our SCN8A community? I think, Steve, you spoke a little bit to that about wins across across communities, but is there something specific about Angelman syndrome that might be particularly helpful for our community?
I think it's the general hub. I think that's the work from Ultragenics, I believe, that was announced just recently. And I think it goes back to that same point that positive results in one area will make, and there are a remarkable number of companies now looking into this. One of the reasons is that Ionis held patents around specific technology to make these ASOs. Those patents have expired, so other companies can now work in that space, whereas before they were denied because of intellectual property concerns. And that's really opened up the vista now. And I think there are many labs and many companies thinking about all these different types of applications of ASOs. And again, I think the Angelman is just another great story that if that works, that we can see an approach for 8A patients.
Wonderful. And I think I'm going to ask one last question before we wrap it up. Although there are more, and I will probably bug both of you with the remaining questions to bring them back to the support group. But I think about one of the big questions we saw pop up in a couple of from a couple of parents was age of treatment. So how could we expect? I know, Miriam, you are looking at the ASOs at different ages of the mice, and I don't know if there's evidence that in other areas that would speak to treatment at a particular age and how much could be reversible, if anything.
In the case of 8A, it's clear that the neurons need 8A for their whole lifetime. If we turn it off in an adult animal, we generate seizures within 2 weeks. I'm sorry, if we turn it up, if we turn the gene on. So my feeling is that it will be possible to have an effect almost at any age. As soon as you get the neuron to stop firing like crazy, I think you'll see a lot of recovery.
That's very exciting. And I think I'm just going to ask one more question from the group here, because Teresa, you're after my own heart, you say dumb question. When these gene therapies, when will these gene therapies be available, and who would administer or treat? So I'm assuming that this is after a clinical trial would be completed. If an ASO became a therapy that was available and run through our insurance, where would we be getting those therapies?
I mean, I can answer that. Generally, I mean, you look where SMA is being delivered, so you normally need some sort of tertiary center, so a university hospital, or a sort of it won't be at a local clinic, it'll be at specialized centers, and clearly in the US there's enough capacity to treat all the SMA children who need it, and there are more SMA children than there are 8A children. So that there's sufficient capacity. And as Miriam said, if the delivery is, every 6 months or so, it's a procedure that takes several hours on the day. And as this becomes available, more clinics will gear up to do this as well. But you'd want to go to a good center.
It might be more like like a step above an infusion therapy of some variety.
Right.
Yeah, because it'll be an injection into the spinal cord.
Yeah.
Similar to an epidural.
Yeah. Okay. Well, on that note, I know we've gone over, and I'm just so grateful for both of you to hang with us for so long and answer these questions. I think this was really was the most active question and answering from our families, and I will pass along those extra questions to you guys, and Steve, if you could forward your slides to me when you have a moment, I can share those with the families as well.
Sure.
And Miriam, I have yours now, so
Great.
I'll share those. I'm just so grateful to you guys for joining, and you can see all the parents are saying thank you. This was really just a wonderful session and so informative. And I love that our families are asking these really deep and difficult questions, and you guys are right there helping us understand them. So thank you. And
Thanks,
thank you,
Hillary,
parents,
for the organizing. Thank you very much.
Absolutely. This has been such a pleasure. And thanks, parents, for showing up, because we can't do this unless you show up and ask questions. So
Absolutely.
And thanks again to Casey and Megan for handling everything on the back end, and to our sponsors, Neurocrine, Praxis, and Zogenix. And I won't see you next week, but we will see you in 2 weeks, gearing up for the gathering. So thank you, everyone.
Thank you, everybody.
Talk to you soon.
Bye-bye.
Bye-bye. Thank you.